Welcome to a quick tour of microfluidics, the science that shrinks entire labs down to tiny chips. In this presentation, we'll explore how controlling fluids at microscopic scales is reshaping diagnostics, research, and industry, and why manipulating the unseen is unlocking faster, cheaper, and more precise ways to solve real-world problems. This slide contrasts the old laboratory setup with the new microfluidic approach, showing a cluttered bench next to a single chip. Instead of days, large volumes, and high costs, microfluidics promises minutes, tiny droplets, and lower expenses, essentially turning slow, resource-heavy workflows into compact, high-throughput processes that accelerate discovery and reduce waste. At its core, microfluidics is about precise control of fluids in channels no wider than a human hair, where laminar flow rules. The diagram shows how turbulent mixing at the macro scale becomes predictable, layer-by-layer flow on a chip, allowing scientists to separate, analyze, and manipulate components with exquisite accuracy and repeatability. Think of a microfluidic chip as a miniature lab made of layered parts, microchannels that act like highways, pumps that serve as the engine, valves that work as traffic lights, and detectors that observe reactions in real time. The exploded diagram helps visualize how these elements stack and cooperate to perform complex experiments on a tiny footprint. When designing microfluidic systems, material choice and channel layout matter as much as the concept. Flexible PDMS enables rapid prototyping, glass provides precision and chemical resistance, and paper offers an inexpensive route for point-of-care tests, while channel shapes, whether wide lazy rivers or narrow water slides, dramatically influence how fluids behave and mix. There are three main microfluidic approaches to know: lab-on-a-chip systems that integrate complex lab functions into a single device, paper-based platforms that use capillary action for low-cost diagnostics, and digital droplet methods that electronically manipulate tiny droplets for programmable experiments, each suited to different needs from research to field deployment. Microfluidics is powering real-world transformations across medicine, pharma, and the environment. Rapid point-of-care diagnostics can detect disease from a single drop of blood, parallelized testing accelerates drug discovery on living cells, and portable sensors enable on-site water and soil monitoring to catch contaminants before they become crises. The advantages are clear. Experiments that once took days can now run in minutes, costly reagents and sample volumes shrink dramatically, and the fine control at microscales yields higher resolution data and more reliable results, making microfluidic platforms a versatile, Swiss Army knife-like tool for many scientific and industrial challenges. The road ahead still has hurdles, from technical sensitivities where a single dust speck can disrupt a micro-experiment to complex regulatory and market barriers that slow adoption. Researchers are tackling these issues by developing more robust, self-contained materials and pushing for platform standardization and clear demonstrations of real-world value. Microfluidics gains strength from powerful synergies with other fields, forming a foundation for future innovation. Paired with nanotechnology, it enables targeted drug delivery, integrated biosensors create portable diagnostic devices, and combining microfluidics with AI accelerates data interpretation and discovery, opening doors to smarter, faster, and more personalized solutions. The future of diagnostics and discovery will be smaller and more embedded in daily life, with at-home monitoring via smart chips, AI-guided automated lab-on-a-chip systems running millions of experiments, and low-cost portable tools bringing diagnostics to underserved regions, fundamentally changing how we detect disease and develop new therapies. Let's bust a few common myths. Microfluidics isn't just a futuristic lab toy, it's already in everyday devices. While initial setup can cost money, long-term savings from reduced reagents and automation are real, and it's more than miniaturizing pipes, it's about harnessing unique microscale physics like laminar flow to achieve results impossible at larger scales. Here's a short glossary to keep you grounded. A lab-on-a-chip integrates multiple lab functions on a single small device. Laminar flow describes parallel, non-mixing fluid layers crucial for control. Microchannels are the tiny pathways etched into chips. PDMS is a flexible silicone used for fabrication, and capillary action is the force that drives paper-based fluid flow. If you want to dive deeper, start with foundational readings such as Jean Berthier's Introduction to Microfluidics, follow structured online courses on platforms like Coursera and edX, and stay current by reading journals like Lab on a Chip and Microfluidics and Nanofluidics to track breakthroughs and practical applications shaping the field. The smallest streams are carving the grandest canyons of discovery, and that revolution continues in miniature. As we finish, remember that tiny droplets and microchannels are enabling big changes across science, healthcare, and the environment, and the next wave of innovation will likely come from thinking small and acting boldly.